Well, hello and welcome, students. Welcome back to your next lab. Remember, I am your instructor, Vanessa. I'm going to go ahead and instruct you how to do our cellular respiration on campus lab, because you've known that we've done some that are virtual and some that on campus. This is the instructional video for the on campus. So you will go ahead and find your lab report, cellular respiration and photosynthesis lab. And you're like, photosynthesis? Well, we'll talk about that a little bit. So let's go ahead and get started. I'm looking at the lab. I'm going to try not to refer to too many specific page numbers, because depending on how your lab prints out, it might throw it off. Same thing goes with the tables. Haven't figured out why, it just does. So I will try to guide you the best I can. So in looking at that first page, always read that safety note. Safety note says, be very gentle with the animal specimens. They're going to be used for the entire week. Okay, so depending on which campus you are at, you're going to have a variety of animal specimens, and you'll find out when you get there. So some of the campuses are going to be mealworms and earthworms. Some of them are going to actually have some roaches. Ooh. Some might have some millipedes, and so just be careful. We're not trying to kill them. We would like them to be alive after everybody gets done with them. Okay, so just make sure to be gentle with them. Post-lab cleanup. Your post-lab cleanup, just make sure that your plants and aluminum foil is disposed of, preferably in the recycling bin that's outside of your doors. I would like that. So as you continue on, you get to see your lab purpose, okay? I'm not going to read this step by step, but what I am going to tell you is the purpose of the lab is to illustrate cellular respiration, okay? Now, cellular respiration is the metabolic process that is taking place inside of your cells. And the outcome of cellular respiration, the whole purpose of it, is to make usable energy called ATP. And ATP is a usable form of energy. So it's actually an energy-carrying molecule, if you want to be specific. So as you look at your figure 1, you can see the cellular respiration. You've already covered this in your lecture. So really this lab should just be something additive to the information that you've already gotten. And if you see the next figure 2 and figure 3, it'll show you your photosynthesis. So go ahead and refer to, go ahead and refer to your first figure 1. I'm going to go ahead and drop the formula for you. You should know this formula because you've already tested on it, but I'll draw it up again. Here we go. So cellular respiration. Just make sure you can see that. Good. Cellular respiration formula. You're going to inhale oxygen. So you inhale 6 O2 molecules. Well, what do you eat? You eat sugar. I should have put that first. I always put that first. Why would I do that? You eat sugar, glucose. So C6H12O6 plus 6 oxygen molecules, O2 molecules, okay, is going to yield waste, and the waste that is going to yield is carbon dioxide, specifically 6 CO2s. So that's 6 carbon dioxide molecules. Okay. It's also going to yield 6 water molecules. And like I told you, the purpose of this is to make ATP. So ATP is going to be the energy that is carrying the energy from the glucose. And so we put plus ATP. Officially, energy is leaving out of this reaction and being sequestered within another reaction to make ATP. But this is the net formula. Now, you'll read in your book that there's actually some waters put in on this side of the equation into the inputs, and there are 12 waters on the actual outputs. So this is your net equation. I'll say it again just in case you can't see. Glucose, C6H12O6 plus oxygen, breathe it in. 6 O2 molecules is going to yield, that is an arrow sign, 6 CO2 molecules that you're going to exhale as waste. Then 6 waters, they're going to be excreted from your cells or used within your cells because our bodies are mostly water. And the whole point is ATP. ATP is a high energy molecule. So that is your cellular respiration formula. So your photosynthesis formula is actually on that paper. It is almost the opposite. It is not the exact opposite, okay? I'll show you why. Photosynthesis. I'm just going to put photo right there. Your plants. Let's go ahead and turn this back around so you guys can see again. There we go. There you go, your photosynthesis. Your plants are going to take in CO2. So 6 CO2 molecules are going to be taken in by the plants, okay? Or could be protists, or could be photosynthesizing bacteria. Alrighty, but animals do not photosynthesize, remember that. And then they're also going to take in water molecules. 6, I'm giving you the net formulas, 6 waters. With that, formula goes in this direction, but this one you need to input energy. So sunlight, sun energy, or some type of energy is going to be input into the reaction. That's why it sits right here on the reactional arrow. And so the byproduct is going to be glucose, C6H12O6, not 06, 06. And you're going to get 6 oxygen molecules as waste. So as you can see in your formulas, they look very similar, but they are not exactly similar. Let me go ahead and turn off that little circle right there so you can see a little bit better. So the reactants of cellular respiration are the products of photosynthesis. The reactants of photosynthesis are the products of cellular respiration. But as you can see, this right here is an endergonic reaction. An endergonic reaction meaning energy, solar energy, is put into the reaction. This one here is an exergonic reaction because energy is going to exit out of the reaction. So they are not the exact same thing in reverse. So make sure you never put that on a test, okay? All right, let's go ahead and get started. I'm going to go ahead and put that light back on since I'm standing in front of that little circle. All right, so back to your lab. So there's your quick little review. Feel free to read your review process there. Before you get started, so I have gone through past figure 4. What we're going to be reading, just go ahead and pass your figure 4 in photosynthesis. It's before you state your hypothesis, okay? We are going to be trying to figure out the respiration rate of our organisms, okay? So we're going to do some animal organisms and we're going to do some plant organisms. Like I told you, depending on the campus, depending on the availability, you don't know what they're going to be yet. So I've already given you a couple of the animal organism examples. Some of the plant organism examples are going to be peas or chickpeas, spinach. And so we already have some peas here that I'm going to show you, for example, today. They would be germinated. That means they're nice and wet and been sitting in the sunlight or some type of light, and they're ready to go and get started in photosynthesis. So with that, from what I've told you, from what you have read, I want you to read through what you're going to do, then come back to the hypothesis, okay? I want you to state a hypothesis before you get here for lab. You need to always state a hypothesis before you begin experimentation. Otherwise, it's backward science, and it's not valid. So, You also want to state a prediction. So the hypothesis is an explanation. Not the only way to state a hypothesis, but I've talked to you about this before. If-then statements are great. They keep it kind of simple. They don't have to be, but whatever is in your hypothesis, you want to see a cause and effect. And a prediction, predictions can get more specific. And so, with that, moving on to the materials. It says you're working in groups of 3 to 4. Really, you're going to be working in probably groups of, it really depends on which campus you're at, most likely. Excuse me, hiccups too. It gives you a list of all your materials. Your materials will be found in one of these buckets, or if you're at the other campus, in the green bucket. Make sure all your materials get back into the buckets, but make sure your organisms are classroom materials and they get back to the classroom cart, okay? We don't want poor little organisms left in the bucket where they're not going to get fed and then they end up dying. All right, so with that, to get set up, so procedure. I'm looking at your procedure part of it. To get set up, you want to set up your LabQuest equipment. The nice thing is you have already used your LabQuest equipment. So this is your veneer. You've used this before several times. You've plugged it into several different things. This time, you're going to plug it in to this, what it is called a carbon dioxide monitor. And so the carbon dioxide monitor is going to be able to detect how much carbon dioxide is coming out of an organism. Okay? So look at that. If we're working cellular respiration and CO2 is coming out of the reaction of whatever organism we're testing, this is how we're going to be able to figure out the levels of cellular respiration of our organism, depending on the amount of the CO2 in that, and it's calculated into a respiration rate, which was on the previous page. So follow the instructions, plug it in. Mine has actually been plugged in for 90 seconds already. Use your pointer, okay? You want to get this set up. If you ever get lost, hit the house button. House button. If you ever get lost, okay? Now when you hit that house button in the middle and you got lost, whoops, let me do it again. Now you always choose LabQuest app. LabQuest app, right there. Boom, you're ready to roll. So from there, you're going to go ahead and set this. It says enter the pointer into the calibration. So we need to calibrate this. As you calibrate it, what you need to do is there's a little hole on this back side. Okay, you're going to place the pointer into the calibration button. If you find you have stuff on here, why don't you empty it? See, I have things. Let me go ahead and erase. May or may not. I have things on there is like in the way. So what you can always do is hit File. I hit File, New, and look at that. The screen changed. So if you're ever lost, hit the house, go to that home screen, that LabQuest app, hit File, New. It dumps all the old data and you're ready to go. So no matter what you're doing, that's how you get there. Alrighty. So I've done that. Now we need to go ahead and we need to, the probe should, hang on, where am I? Oh, we have this thing called our respiration chamber. I'm just following your instructions here. This respiration chamber, you're going to put the CO2 monitor in the respiration chamber, just like such. So as you're getting ready to calibrate, what you're going to need to do is look at your sensors, look at your monitor, plug this in, okay? As you can see your monitor, you can see the file and sensors. Hit sensors, working upside down here. Now hit sensor setup. Look at that, sensor setup. So right away, it tells you where to choose. So we're looking at the CO2 gas. It doesn't matter if you're in channel 1, 2, or 3. It'll just choose a channel, okay? Whatever channel you're in, and hit OK. And then we're going to go ahead and calibrate. So in order to calibrate, place these in the chamber. Trying to show you everything all at once. Luckily, you have somebody else and you put your hand on the table. You're going to hold this in here and hold it until that red light on the side is going to stop blinking, and it's going to hold hard red. About 30 seconds. I have to look at it. There we go. Perfect. Now I'm looking at my parts per million on mine, and you're trying to get about 380, okay? It's going to toggle. It's not going to stay 380 all the time. I'm just letting you know that. If it toggles somewhere around 400, 420, or 320, something like that, you're good, okay? So with that, this is nice and calibrated. Don't unplug it. The moment you unplug it, you're going to have to recalibrate it. All right, so next, you need to determine the mass of your organisms, okay? So some groups are going to be working, actually, I haven't decided, it really depends on which organisms, but plants and animals will be worked with. Because plants are so easy to work with, all you have to do is measure the mass of the plants using a weighing dish. And so you make sure that your that your electronic balance is turned on. Just go ahead and let it stabilize. It'll usually count down, let you know when it's stable. Place your electronic dish on your electronic balance, your measuring boat or dish or paper. Now let's go ahead and zero it. You hold the 0 or the tare button. This is stuff we've done already. Okay. Something happened and I turned it off. I held it too long. Look at that. Let's get this ready again. Put it there. Let it stabilize. Hit the 0 button. Perfect. Now, you get your plants. Place them on. We'll just let those ones stay on the floor. Place it on your dish. Write down the mass of the plants. Okay? Great, all done. Make sure you write that down and you keep the amount that you have written down. Now, animals are a little bit harder because animals like to wiggle around. So, this is what you have to do with the animals. You have to have yourself an empty respiration chamber. You take that empty respiration chamber, okay? Go ahead and get the mass of your empty respiration chamber. Now, if your animals are cooperative, you could probably just put it in there and go ahead and get the difference, tare it, put it in there, and then get your mass. Yeah, sometimes the roaches, sometimes the crickets, they do not cooperate. So what you're going to have to probably do is get the mass of the chamber by itself, okay? Get your roaches and your crickets, get them in there. Try not to let them out. Try not to push on it. And then get the mass of them with them in there. And then you calculate the difference. So I'll let you work that out. It's real fun. All right. Next. So once you have the mass of your organisms, you're going to go ahead and put the mass of those organisms in your table 1 results. So I'm looking at your lab report. Alrighty, so with that, and if it tells you to go ahead, if you just did the mass of the organisms but you didn't do the actual difference, that's fine. Just leave that part blank. Now, we're going to go ahead and expose our organisms to different conditions. We're going to expose our organisms to conditions of light and expose our organisms to conditions of dark, okay? And that's where the photosynthesis piece is going to come in. So, as we do that, first thing we're going to do, here's what I say. I don't care what the order on here says. I say do the dark first, okay? I find what happens when you put certain organisms, certain animals in the light first, they get very stressed, and then they don't respirate when you put them in the dark. You almost think that they died, okay? So I always say put them in the dark first. So here's how you're going to do this. I'm only going to illustrate with my peas. Just know you're going to have to do this with your peas and your animals, okay? So that's why this is a time-intensive type of a lab, so that's why you're watching this video first. In order to illustrate a dark situation, you're going to get some aluminum foil, just like such. Just wrap it around, make it dark. That way no light gets in. Now, you're going to put in your respirometer, and then you're going to run your respirometer for 3 minutes. So I've shown you in the past how to set the time, the duration on it, but if you're not familiar with it, just go ahead and look to the right side. It has duration. You can go ahead and set that for 3 minutes. So I'm just going to go ahead and set that for 3 minutes. 3 minutes is 180 seconds. It defaults at 600 seconds. So there you go. Now, here's the important thing. You have to hit the play button, okay? So when you're doing this, in the bottom corner, there is a button. There is a play button. Hit the play button. There you go. When you hit the play button, you're going to see a graph build. And this is going to go over the course of 3 minutes. So you keep your respirometer, your CO2 sensor inside your respirometer with your organisms for 3 minutes. And you just let it play. Okay? So I'm just going to give it away. These peas have not been germinated yet, so my value is probably going to end up 0. But yours won't. Whenever you come in, yours will be germinated. And you're going to start to see a graph grow. Now, what this is doing, it is figuring out the respiration rate, the parts per million of CO2 gas that is taking place or being excreted over time. That is what the machine is doing. It's doing that for you, that way you don't have to do that. But you will have to use that in order to calculate that next step. And just hold on to that, I'll explain that. So don't hit the stop button, but at the same thing, I don't want you to sit here and stare at me for 3 minutes, so I'm going to hit the stop button just so I can show you what's next. Oh, wow. Isn't that beautiful? Look what I got. Now, this is not my final value. You're going to follow your instructions on your in your lab report, and it's going to tell you to hit analyze. Do you see where it says analyze? I know it's kind of fuzzy. I'm trying to stay close, but not too close. Hit analyze. There you go. And then you're going to hit curve fit. So these instructions are in your laboratory, and then you hit CO2. So when you do that, you're going to choose a coefficient. So there's a drop-down menu over here. In that drop-down menu, you're going to choose linear. So choose linear right there. Nice. So look at that. What it did, it actually made the graph of a line through that graph of time that you ran your CO2. So here's the values that you're going to write down in your lab report. You are going to write down M. M is the slope. Do you see it? M. You want the slope. So that is the slope of the line. The slope of the line is the rise over the run. Remember that from your algebra. All right, so where do you write this information? Let's go ahead and take a peek. Oh, by the way, pull this out. Let your organisms breathe while you're writing everything down. That is crucial, okay? That is so crucial to do. So you are going to write down the slope in table 2. You can even see your chamber 1, animal in the light. Chamber 2, animal in the dark. Chamber 3, plant in the light. Chamber 4, plant in the dark. Right now we're doing plant in the dark. We wrote down the slope. Then Once you get all that done, you can let it breathe. And this is really comes in handy. Provide it some oxygen. Why would I provide my organism some oxygen? Well, because in cellular respiration, you need oxygen. That is why. Give it a little oxygen. And then clear your machine. You just hit File, New. Remember how I showed you in the beginning how to dump your files? Go ahead and discard it. Do it again. Boom. Run it. 3 minutes. Same exact thing that I just showed you. And then go through the process of analyzing your data and getting the slope, which is M, and you're going to write it for slope number 2. Then, while it's sitting and waiting, and you're doing your writing, your group members giving your organism some oxygen, especially if they're animals, okay? And then you do it a third time, okay? After the third time, you can go ahead and take the average of those 3. You might find that your slope is going negative. Don't worry about it. You're going to figure out the average slope for all 3 trials, and you're going to put that in the last column. All right, so next, you are going to do the exact same thing. I would probably just do it immediately, same organisms, in the light. So I just did it in the dark. And this is not enough light. We are going to have actual spotlights. So I have brought this little tiny wimpy light just for illustration, but we are going to have lights that are going to be very, very powerful. So make sure you put some water. This is a beaker of water in front of your light. Okay? So that way when you turn on your light, you want to put your organism directly in front of the water. Because if you recall, water has a very great ability to absorb high amounts of energy without having large changes in its own temperature itself. All right, so you're going to put your organism in the light. Make sure you clear your data. File, New, discard, 3 minutes, do it all over again. Okay? Then once you get that data, you're going to write it on the next line, plant in the light. I said, remember, go in my order. It doesn't matter that order. I say do the dark first, then the light. That's just how I work. Alrighty. So then you do this 3 different times, the exact same way I explained before. Take the average. All right, friends. I'm going to go ahead and get rid of this light. You'll do all of those same things for your animals. You'll do your animals in the dark first. Then you'll do your animals in the light. 3 trials of the animals in the dark first. Then the 3 trials of the animals in the light second. Okay? We're not intermixing, just making sure you're getting all of them done first. Now, crucial with those animals, they need to breathe. And especially with the heavy, heavy lights that we have, they're going to get stressed. And you're going to actually see that in their respiration rate. So I guess I'm giving you some clues, aren't I? Alrighty, so once you have done that and you've collected your data, you then need to figure out the average respiration rate. So I'm now looking at your table number 3. Table number 3 will be the average respiration rate for your organisms. And so as you can see, the organisms, animal in the light, animal in the dark, plant in the light, plant in the dark, seeds in the light, seeds in the dark. I'll let you know, depending on your lab, what we'll be using. The mass of the organisms. Remember initially I had you measure the mass of each of the organisms. So you will take that from your table 1, transfer that information there, the mass of those organisms. Then you will take the average slope, that last column from your table 2, and you will transfer it to that second column of table 3. Using the mass of the organism and the average slope, you are then going to calculate the average respiration rate. And the average respiration rate is just literally the average slope, which is parts per million per second. Parts per million is the measurement of how much gas is coming out, the CO2. Per second is time. divided by the mass of the organisms. Okay, so that is your purpose of this lab, to figure out the average respiration rate of these organisms. Now, we've been working a long time on graphs, and through all of these videos, I've been giving you lots and lots and lots of hints about graphs. Okay, so I'm not going to tell you what kind of graph you're going to make on here, but I will still give you a little guidance. You are going to be graphing that last column, the average respiration rate. Because you are graphing the average respiration rate, are we graphing over time and doing a line graph? No, it's already figured out in the average, right? Are we taking several different data points and using those data points to figure out some unknown data? Nope, we're not doing that either. So since we are taking average values, animal in the light, comparing it to animal in the dark, boom, boom. Plant in the light, comparing it to plant in the dark, boom, boom. I'm going to let you guess what type of graph you're going to be go ahead and giving me. I know I give way too many hints. I know I do. So please answer all of the questions accordingly. This is such a fun lab, but I need you to be prepared when you get here because it is time intensive. Be prepared to start right away when you get here, okay? Now, if you recall, in the beginning of the lab, I told you we were going to discuss how cellular respiration is going to be illustrated through the CO2 and the respiration rate. So that is what we have done. But I also told you at the same time we are going to illustrate photosynthesis. And so this is a thinker, but I bet you once you think about it, it's going to make a lot of sense. Let's talk about the plants. With the plants, did you find that when your plants were in the light, they had a high respiration rate or a lower respiration rate in conjunction when they were in the dark? If they started using the excess CO2 in the chamber when they were in the light, that is an illustration of photosynthesis. So not only are you illustrating the cellular respiration with the CO2 being released, you might find in the dark the plants had a higher CO2 release versus in the light they might have had a lower CO2 release. Why? Because they're also participating in photosynthesis at the same time. So since they are participating in photosynthesis at the same time, they're uptaking that extra CO2 and using it in the chloroplast in order to do the process of photosynthesis. So, all right, I hope you love it.